She is the Centennial Professor of Pediatrics and the Chief of the Division of Genetics and Genomic Medicine at Washington University School of Medicine in St. Louis. Dr. Dickson has studied the pathogenesis and novel therapy development of mucopolysaccharidosis and related disorders for more than 20 years. She has served on multiple advisory boards for the design of clinical studies of brain-directed therapies for MPS disorders. Her honors include the National Institute of Health National Research Service Award Fellowship and the Richard Weitzman Memorial Award for Meritorious Research. She's been elected to the American Society for Clinical Investigation and the Association of American Physicians. Welcome, Patty.
Thank you so much, Scott. Thanks, everyone. So I'm going to talk about mucolipidosis, and I've pressed the wrong button. Here we go. These are my disclosures. So mucolipidosis types 2 and 3 can be divided into these three categories: ML2 or ICEL, which is an infantile or even prenatal onset of the condition with cognitive and physical impairment. And this is a rapidly progressive condition. ML3 alpha beta is a childhood onset with really no cognitive impairment, but physical disease that's mainly skeletal and is slowly progressive. And then ML3 gamma, which is also a childhood onset, potentially a little bit later, no cognitive impairment, physical disease, and slowly progressive, similar to ML3 alpha beta. So how does mucolipidosis cause disease? So we've talked a lot about MPS and maybe not so much about ML. So the MPS disorders are caused by deficiency of an enzyme that lives inside the lysosomes. So the lysosomes are like the stomach of the cell. They digest materials that our body makes or ingests and inside the cell, and then turns them over into other parts to be reused. ML is a little bit different. It's caused by a deficiency of an enzyme that lives in the Golgi apparatus. So our cells make proteins, right? We all have DNA that has genes inside, and the genes are like recipes to make a souffle or a cake. So your protein that the gene recipe makes is your cake. And then as you go through the parts of the cell that make that cake, in the Golgi apparatus, you decorate the cake with little sugars, like icing your cake. And one of those sugars is mannose-6-phosphate, if that cake is supposed to go to the lysosome. And the mannose-6-phosphate on our protein residues binds to a receptor called the mannose-6-phosphate receptor, allows those proteins to go into the lysosome where they work to break down the materials that the lysosome needs to ingest. Without those enzymes, the lysosomes fill up with storage. So that enzyme has a really long name, glycnic-1 phosphotransferase, or you could call it PTAs for short. And it puts that mannose-6-phosphate on again, so those enzymes go decorated into the lysosome. The PTAs has three subunits in it. One is an alpha subunit, one is a beta. I'm sitting here like pointing at the screen as if you can see that. Alpha subunit, beta subunit, and then binds to it is a gamma subunit. These alpha and beta subunits are encoded for by one gene, GNPTAB. The gamma subunit is encoded for by GNPTG. And this is just a cartoon kind of representing how this comes together inside the Golgi apparatus. It sits in the membrane there. So what is S1S3? Stuart Kornfeld's laboratory, including Lin Lu and others, developed a modified version of the normal or wild type PTAs that's a little bit smaller but contains the pieces of the alpha and beta subunits of the PTAs, and doesn't bind to the gamma subunit. So here is a cartoon on the bottom here depicting the sequence of wild type or naturally occurring alpha and beta subunits of these PTAs. Naturally, we make alpha and beta subunits with the one gene, like I mentioned, GNPTAB. These are then cleaved and separately into alpha and beta. With the S1, S3, you have the pieces of it, but not all of the pieces. Makes a smaller protein, makes a smaller gene. That smaller gene is actually easier to use in things like gene therapy because you do care about how much gene you have to put in to the vector. And then that protein is made with the alpha and beta subunits fused together and doesn't bind to gamma. So the first thing that Stuart Kornfeld's laboratory did was look at this S1, S3 versus the normal or wild type PTAs when you put them together with lysosomal enzymes to see how much manna-6-phosphate you get with the regular PTAs versus the S1, S3 phosphotransferase or version of the PTAs. And so this experiment used a percent of glycan phosphorylation to demonstrate the amount of manna-6-phosphate on the enzymes. On the left here, this is alpha-mannasidase. The second one is acid-alpha-glucosidase. This is GLA for Fabry disease. This is GBA for Gaucher disease. So, and acid-alpha-glucosidase is for Pompe disease. Anyway, 4 different lysosomal enzymes showing that with the S1, S3, we see an increase in the manna-6-phosphate content versus the naturally occurring PTAs. Now, how might this work as therapy for ML2 and 3? So in ML2 and 3, because the lysosomal enzymes come out of the Golgi without that manna-6-phosphate decoration, the enzymes don't go to the lysosome, like I mentioned, where the storage material sits. They can't bind to this manna-6-phosphate receptor, and instead they spill out of the cell in large amounts. With S1, S3, we postulate that we can add the manna-6-phosphate sugar decoration back onto the proteins, the lysosomal enzymes. They will traffic into the by the manna-6-phosphate receptor, and they can exit the cell with that manna-6-phosphate decoration on them. That will allow them to cross-correct their neighbors, so go to the other cell, bind that manna-6-phosphate receptor, and then enter those lysosomes to correct the storage in the neighboring cell. By this means, you wouldn't necessarily need to treat every cell with S1, S3, just enough cells so that the decorated proteins can exit and cross-correct their neighbors. We treated ML2 mice with the S1, S3 phosphotransferase. We used a gene therapy to deliver it. I'm showing you here 3 different lysosomal enzymes, and this is the same group of ML2 mice or wild-type mice looking at their blood and how much manna-6-phosphate is on their lysosomal enzymes. So beta-hexasaminidase, alpha-mannasidase, and beta-mannasidase were the 3 lysosomal enzymes depicted here. So shown in gray, this is the normal amount of enzymes that are in the blood and that have manna-6-phosphate on it in the wild type or normal mice. In black here are the ML2 mice showing that they don't have manna-6-phosphorylated lysosomal enzymes in their blood. But following treatment with the S1, S3, either 2, 6, 10, or 14 weeks after treatment, we see high levels of manna-6-phosphorylated lysosomal enzymes in their blood. We crossed the ML2 mice with a type of mouse that has a tag on their lysosomes, which allows us to pull down the lysosomes and study what is inside. So this is a flag tag, what they call lysosomal immunoprecipitation, or lyso-IP. And so we have this mouse now for study. we treated them with the S1S3 phosphotransferase gene therapy. This is a graph showing the lysosomal content of brain lysosomes in the mice that are ML2 or ML2 and treated with S1, S3 phosphotransferase. And so shown on the y-axis here are the brain lysosomal enzyme amounts. So the lysosomal enzymes that are in the lysosomes as a percent of wild type. Black bars are the ML2 untreated mice. The, what color is that, aqua? Aqua bars, hope you can see that, are the ML2 mice that had been treated with gene therapy, showing that there was some increase in the lysosomal enzyme content of the brains with all of the enzymes that we assayed in the treated mice. Overall, the mean of the untreated ML2 mice was that they had 47% of wild-type enzymes in their lysosome. In the brain, in the treated mice, we saw that at 73% of the wild-type amounts. So what are our next steps in S1, S3 development? So the first is vector design optimization. So when we talk about gene therapy is where we're injecting a therapeutic into a patient that involves a viral vector. This vector enters cells, it corrects the cells and then secrete protein, and the secreted proteins cause cross-correction, as I showed in my previous cartoon. This is a schematic of what gene therapy looks like. This was not our work, but it was a nice review. This is AAV, or adeno-associated virus, which is a very commonly used gene therapy. Does not cause disease in people, but is a great way to deliver genetic material. So it has a capsid on the outside. Inside, there is the genome of the virus. Again, no infectious elements, but just things that can help put that gene that you want into the body. This cartoon below shows how the gene therapy goes into a cell and then goes into the nucleus where the DNA is. It sits outside of your own chromosomal DNA, makes its own recipe book, if you will, to generate its proteins that are then made by the machinery of the cell. So we are going to be working to determine which capsids, which promoters are ideal to optimize the delivery and effectiveness of S1, S3 in our preclinical testing. Another next step that we plan to perform is human cell testing. And so what this will involve is testing S1, S3 in cells that have the same genetic variants patients have. Remember in the cartoon, we saw that there were alpha, beta, and gamma protein for the PTAs. There's 2 alphas, 2 betas, and 2 gammas. We want to make sure that the genetic variants patients have when combined with the S1, S3 don't cause any protein aggregation that might cause any kind of safety events. So we want to study in S1, S3, or lack of efficacy, we want to study S1, S3 in the ML3 alpha, beta, and gamma cells particularly. There's the cartoon. And finally, we're planning a dosing study in the feline model. There is a naturally occurring cat animal model of ML2. It shows facial changes, small size, neurological disease. It has a shortened lifespan and physical disease in the lungs, heart, and bones. And so we will do a limited study in cats just to determine whether we can see the effectiveness of the S1, S3 gene therapy in this model. So this is our team. I am working with John Wolfe at Penn, and who is also at Children's Hospital Philadelphia. Balraj Duray, who's a research scientist formerly with the Kornfeld Lab, knowledgeable in S1, S3, and lysosomal trafficking. And I want to put a call out to, we need your help. I know that we've been working with Jenny Jackson and Scott to plan natural history studies. We need your input and advice, and we need funding for this project most of all. And I want to acknowledge we have had a really good track record of funding to get us to this point. We were fortunate enough to get an NIH grant from NINDS, or National Institute for Neurological Disorders and Stroke, that supported the mouse work that we've done to date, the Yash Gandhi Foundation, and from the National MPS Society. Much help from M6P Therapeutics and from the WashU IDDRC. I also want to acknowledge Stuart Kornfeld, whose development of S1, S3, and all of his work in ML and the PTAs made all of this possible. We lost Stuart last year. Thank you very much.
Thank you very much, Patty. And I want to also make a shout out that we have provided a challenge grant to Patty, although we still have to raise a lot of money to support that challenge grant. So we're asking to raise, or trying to raise $500,000 to support her pre-IND work to get this into the clinic and into patients as quickly as we can. So please support us in any way possible that you can. And I open the floor up to questions.
Patty, very nice presentation. I was just intrigued by your lysosomal capture experiment, where you saw lots of residual enzyme activity.
Yes.
What do you think is driving the disease pathophysiology when you still have 5 or 10% residual activity for at least even some of the things like IDS and ironidase? What's driving the disease in those ML patients?
It is a fabulous question. There is quite a bit more enzyme activity in the brain lysosomes than we anticipated. And I think it could speak to a number of questions. One would be a question of, are there other transport mechanisms that we have yet to identify that might be at work in brain or elsewhere to transport lysosomal enzymes? Is there spillover? Our tag is a flag tag that is bound to lysosomal-associated membrane protein 1. Maybe that protein might have other endosomal compartments. Maybe there's some more to be discovered about what actually LAMP1 binds to, although we do perform studies to make sure that we're not capturing other parts of the cell, other organelles with this tag. But yeah, I think that and we're not the first to make the observation, as Rich is here, probably has something to say about it, but there is more in the lysosome than we thought in the brain. I have not looked at any other organs.
But just in the brain, it's just either, as you allude to, there may be something else going on, or you're pulling out something down. It's just, it opens the question of why. And
Yeah.
I think it just shows us what we don't know yet about the pathophysiology. Thank you.
Yeah, absolutely.
Yeah, it was great to see the lyso IP and have that in there. I think obviously this enzyme S1, S3 works better. I think one other big advantage is it doesn't require that cleavage step, which is an advantage. I mean, could be a rate limiting step. We know that the normal enzyme, not all of it gets processed. It has a very complex maturation process, so I think that's a big advantage. On the flip side, you're doing so well mannose-phosphorylating these enzymes, especially the ones that end up out in the bloodstream. Is there a concern that because they're so well mannose-phosphorylated, they're going to be too rapidly cleared by the liver? And I know that's a hard thing to determine, but it doesn't seem. I mean, if you're getting it into the brain, it's increasing its levels. But just wondered if you had perspectives on that.
Yeah, I'm not sure how we would tease out the viral delivery to the liver. sink versus the enzyme delivery to the liver sink? I mean, it's a question I hadn't really haven't thought about. We do, the first graph was serum lysosomal enzymes in the mice, and they are still very elevated in the serum, regardless of any liver sink. But one of the things that we're hoping to study with vector optimization is whether we can deliver kind of more broadly and avoid that, at least with the gene therapy component of it. I do think that, as we learn more about how to deliver therapies in general, I mean, this is a disorder where it's going to be a big challenge because we're trying to deliver to bone, to joints, to kind of the whole body, and then into the brain, we think. We think that there's a cognitive component to this, although I mean, I agree with Joe. I don't know how well that's really been studied or how well it can be studied in ML2.
Hi, Patty. Sort of a follow-up question to the issue that Joe brought up. Is there, I mean, as I understand it, LAMP1's going to be expressed on Golgi surface because that's part of the process of getting it to the lysosome. Is there a possibility that you've got contamination of Golgi vesicles in your pull-down that you're using with LAMP1 using the magnetic approach? And could that explain some of the high levels of enzyme and what you're finding?
We can look at that. I don't think this, I mean, this is a tag that's used by others as well. It's not been an issue, but we can look at that.
Just curious, because it's anomalous.
Yeah, no, I agree.
Do we have other questions? All right, if not, I give a shout out to Patty again and thank you.